and for today we're going to be talking about the adaptive immune system. Now, before we get into a deep discussion about adaptive immunity, one thing that I wanted to just touch on was the ways in which we can become immune. Most of the things that we've discussed up until this point have to do with going out and about in the world, running into a pathogen in some kind of forum, and then becoming infected with it prior to the immune response kicking off. That is a very valid way to become immune to something, but we have other kinds of immunity as well, like different kinds of natural immunity or those that are induced artificially. And these are important just to kind of set the stage for us going forward. Natural immunity can come around in two main ways. We can have active immunity, which is kind of what we've discussed, where you go out somewhere, you contract some kind of infection, and your body is going to mount its own immune response to it. There's also the passive immune route, where you're still getting it in a natural way, so not something synthesized in a laboratory, not something prepared, but something that was made by another human that gets transmitted to you and confers immunity unto you as a result. A great example of this is through breastfeeding. Many aspects of a small baby's immune system are bolstered by breastfeeding from a mother. In situations where a mother can't breastfeed, the immunity is going to have to come from other parts of life, like babies putting everything in their mouths or hanging out with friends eventually, or even to a great extent through some of the foods that they consume. Artificial immunity allows our body's natural responses to still go through their own motions, but the way that those pathogens or antigenic features are introduced to our systems is done in an artificial way. So, for example, one that we're probably all very familiar with is vaccination. So by taking aspects of a microbe or a pathogen and introducing them into our system, still gets our bodies to undergo all of the different steps to mount a proper immune response to fight that infection off and create that immune memory, which we'll talk about in detail today, but without the risk of actually getting the infection and potentially the disease state that comes along with it. So what a vaccination essentially is that primary exposure to a pathogen without actually having to get sick with the pathogen. So it's a very effective way to protect people and prevent them from all of those negative consequences that disease state can confer upon them. There's also the passive way to receive artificial immunity, which is similar to breastfeeding, except in this case, we're receiving preformed immunity in an artificial manner, so through an intravenous, for example. But no matter which way you slice it, all of these methods will allow you to develop immunity against something that is immunogenic or antigenic. We talked a lot last time about the innate immune response, all those physical and chemical defenses that are in place to protect our epithelial layers and our common portals of entry from being breached. But one of the most important things to remember is that the innate immune response doesn't allow our bodies to create memory. So anytime we get any infection, even if it's the same infection over and over again, we're always going to go through those same motions, producing mucus, having macrophages be at the site of infection to destroy whatever happens to be there. This is not ideal, especially in a world where microorganisms are constantly adapting to us. And they are very good at circumventing a lot of the mechanisms that we have in place. So we also need a bit of an edge. The adaptive immune system is what is going to help us to be able to fight things more effectively. The innate immune system, when it fails, will activate the adaptive immune response. We talked last time about how the innate response is very fast. It happens right away. When that does not completely clear an infection, that's when the adaptive immune response becomes activated, and it's almost like calling in the reinforcements. The adaptive immune response is mediated by a specialized cell set. So similar to what we talked about last time with the innate response, there's a separate set of cells that are a lot more specialized that are going to come in and do some of this work. They will actually recognize and destroy invading pathogens and then also remember the pathogen. So if you were to get reinfected later, our adaptive immune response has already determined what kind of infection it is. We talked about this a bit last time. Is it an intracellular infection, something like viruses or intracellular bacteria, or is it extracellular? Because there are two very different sets of cells that will get activated in either of those cases. And so by knowing what a pathogen is and how it's going to actually try to manifest into a disease state, our bodies can deploy a response that's going to be effective versus this experimental phase where it's still kind of trying to figure things out. And sometimes that immune memory is so good that if we do get reinfected with something, we take care of it so quickly that we don't even know that we had an infection to begin with, because those microbes get destroyed so fast, they don't have an opportunity to even cause symptoms. Think about living in a household with many other people. Everybody might get sick from the same infection, but you're not going to see that infection keep going around and around and around forever. Eventually, everybody gets better, right? And people move on with their lives. And this is greatly in part due to that immune memory, which we'll talk about a lot today. Now, the recognition aspect is highly specific. So where the innate immune response will say, well, that's non-self, let's make mucus. The adaptive immune system will say, okay, that's non-self, but what else is it about the specific pathogen that makes it immunogenic? Let's talk about that. It'll also be able to identify cancerous or infected cells and destroy them. And as I was kind of alluding to earlier, the response is specific. So it's not just a generalized blanket response. Based on what kind of pathogen it is, what kind of mechanisms it has, your immune system is going to kick into one of several different branches that are going to be specifically targeting that type of pathogen. And of course, you're going to have that memory aspect, so that if you do have a secondary infection later on, your body knows what to do with it. The adaptive immune response is able to identify a diverse number of features. The innate immune response will say, well, there's some peptidoglycan. That's not normal. Hosts don't normally make peptidoglycan. Peptidoglycan is something that's common between most bacteria, right? But the adaptive immune response will look at a cell and say, okay, well, that's non-self because there's peptidoglycan in it. But hey, there's a mannose receptor on the surface of that. That's not normal. Our host doesn't produce that. Or it'll say something like, oh, there's a transport protein that's used only in bacteria. That's not produced in the host. So it'll recognize very specific antigens on the surface of cells versus just these broad features that are common between most types of bacteria or viruses. There's also a specificity to this as well. So if you have a single protein that has hundreds of different amino acids, there are a ton of different little three-dimensional pieces of that can all be recognized individually. So imagine the human face. Maybe one part of our immune system will recognize the eyes. Another part will recognize the nose. Another part will recognize the cheekbone. So you have many different features of these antigens that can be specially recognized and targeted by our immune system. So you not only have one set of cells that target one type of protein, you'll have maybe 10 different cells that target a single kind of protein. And that specificity also helps in the case of microorganisms that mutate or change often, because they can't change everything. They can change some things, but then the parts that do not change and our immune system has seen before are remembered. So that specificity really helps with retaining that immune memory and being able to respond better with subsequent infections. In terms of the cells that are responsible for this immune response leg, it is again going to be our white blood cells, or our leukocytes, that will carry out these events. We have two different branches of immunity that we're going to talk about today. There's humoral and cell-mediated. Humoral immunity is what we commonly think of when we think of immunology and the immune system, because this is the leg that produces antibodies. Antibodies are proteins that get produced by B cells, and the antibodies themselves that get produced are specific for all of those little antigens or epitopes on the surfaces of their targets. This is the primary response against extracellular infections, so things that occur outside of host cells. Extracellular bacteria, it could be toxins that get produced by those that get secreted into our systems, or even in some cases viruses during the lytic phase where they get released from our cells and are in that extracellular space. Antibodies get circulated through our blood, so they can only see stuff that's also going to be outside of cells. They're not going to go knocking on every cell door trying to get in. The other aspect here is cell-mediated immunity. This is done by T cells that are going to act against foreign entities, but that typically have two major roles. They're going to either activate other immune cells, so they're going to sit at the top with their megaphones and they're going to delegate tasks to other immune cells and activate them, or they're going to actually go out and primarily respond against infected cells, so intracellular infections. things like viral infections or intracellular bacteria, as we've talked about. And this is done through a mechanism where our host cells will be able to kind of send a little bit of a message to the outside world with a signal to say, there's something going on in here. You probably heard the blink twice if you're in danger for someone who can't say something out loud if they're in a serious situation. It's kind of like that, where cells can't necessarily take care of the problem themselves. They're kind of in a hard place, but they can still signal to the immune system that something is wrong. This diagram very nicely summarizes some of the things that we're going to be talking about today. So you can see our humoral immune response. Here are some extracellular microbes like we've been mentioning. These are our B lymphocytes, or our B cells, that are going to produce antibodies. You can see those little Y-shaped proteins that are being released from these cells that can then go and bind to all of those pathogens, and those act as opsonins. We talked about that last time. They are an adherent flag that's going to be attached to those features of those bacterial cells and let our immune system know that they need to be destroyed. In the case of toxins as well, they can also block a toxin's activity. For the cellular immunity, you can see up at the top here that we've got a cell that is infected. This also applies to cancerous cells. And then we have other cells that come along. They see the antigen, they see the infected cell, and then their job is to actually go along and either activate other immune cells or come directly to the infected cell and destroy it. So when you can kind of see it like this antibody response isn't really going to be effective against intracellular pathogens. And this intracellular pathogen response isn't really going to be effective against extracellular ones. So the way that our immune system is activated is really important. And the fact that our body remembers which pathway to take is also really important and helps speed up the process in subsequent infections. So the B and T lymphocytes are produced in our lymphoid organs. Our primary ones, you'll recall that there are two primary ones, bone marrow and our thymus, and everything else that's affiliated with the immune system in terms of tissues and organs is considered to be secondary. So T cells are formed in the thymus, B cells are formed in the bone marrow. That's pretty easy to remember. So what we have circulating in our system at all times are B and T cells that have never seen the antigen that they are made to recognize. You have to understand that the receptors on these cells are specific for something that maybe we've never even been exposed to yet. So these cells are considered naive. They have no experience in fighting an immune response. So they're always in circulation, wandering around, waiting to find what they are made to detect. And that's a pretty neat concept. We have proteins in our bodies waiting to run into stuff that they may or may never meet. But if they do, they're ready. As soon as they encounter an antigen that their receptors are specific to, they get activated. So they see that infection and then they turn on through a variety of different steps, which we'll talk about, and then they're able to start reacting and fighting that infection. When B cells get activated, they're going to start to produce and secrete antibodies, which go off and opsonize and target things for destruction. Helper T cells, which are one of two types, are going to start to produce cytokines in a great number, which can then go and activate other cells, activate different processes, help cells to migrate to certain parts of the body where the infection might be present, a number of different things. They're kind of at the top. And then we have cytotoxic T cells, which are the ones that, when they're activated, are going to go and start to destroy infected or cancerous cells. We're going to talk first about the cell-mediated response, which involves those two types of T cells we just mentioned. We have our cytotoxic T cells and our helper T cells. T cells can only determine when something foreign is present in the body when it is shown to them. So imagine that they don't have the ability to see things when they are completely together in their native states. If there's a bacterium that walks straight past them, they cannot identify it as foreign. What they have to do is have little bits and pieces of that particular entity presented to them. Antigen presentation is done by a number of different phagocytic cells, like we talked about last time. Macrophages, dendritic cells, even B cells in some cases, will be at the sites of infection in the innate immune response, engulfing different bacteria, digesting them, and then migrating away to these sites where our T cells are present, and then they just stick out this little signal and show them a little piece of it. That's how these cells get activated. Once one of these T cells sees the antigen and recognizes it, that fragmentation of our pathogens into these small bits and pieces to show to T cells is done through the normal phagocytic pathway like we talked about last time, where you have a pathogen that gets engulfed, it's brought into the cell, there's a fusion of that phagosome with the lysosome to cause degradation of that material, and that's the stuff that's going to get loaded onto a little protein that's just going to get stuck outside of the cell membrane. The little arm that sticks out of the cell membrane with that little piece of antigen stuck to it is called a major histocompatibility complex. That's a very long name just to say it's a little molecule that holds an antigen. And think of that like the key that fits into the lock of the receptors on T cells. So they put out their little signal, and then our T cells come along and attach to it. And if they recognize that signal, then they can become activated and go along and start to fight an infection. If they don't recognize it, there's another cell somewhere in our repertoire that will recognize it and will become activated eventually. So this is a great diagram that kind of shows those types of interactions. We have an infected cell in this case that has a lot of viral particles inside. They're being degraded and destroyed as that cell is trying to fight off an infection. And here is that MHC molecule that we just talked about. That's that little arm that sticks out from this cell with the antigen bound. And a T cell can come along and bind to this and recognize it. And if it recognizes that, then it can do its effector function. In this case, this is a cytotoxic T cell, which sees this intracellular signal, right, from an infected cell. So it's an intracellular infection. And now this specialized cell can go and start destroying other cells that are showing this same signal, because this signal is suggesting that this cell is infected. So this cell can then go and find all of the neighboring cells that are also infected and destroy those before they are lysed, because lysis, of course, is just going to release more viruses and allow them to spread. Here, we have a mechanism to identify and destroy those cells prior to lysis. That's done through apoptosis. Helper T cells are usually going to be activated by extracellular infections. So you can see in this case, there's the phagocytic pathway. We're having all of these pieces that are degraded through lysosomal degradation. We have the MHC molecule that's going to bind antigen. That's going to get shown to helper T cells. And then these cells are going to be, again, those big delegators. They're going to say, okay, well, this is an extracellular infection. I'm going to push macrophages to go around and engulf more microbes. I'm going to push B cells to start producing antibody. And they activate a number of other cells as well. But like I said a while ago, without this presentation mechanism, T cells cannot see an infection. If this microbe walked right on by this helper T cell, has no idea that that's a problem until it's presented to them in this fashion. So the mechanism for cytotoxic T cells is that they're going to interact with an infected cell, just like we saw in the previous slide. They're going to determine that there's a foreign antigen present, and when they become activated, what they do is they induce apoptosis. Apoptosis is programmed cell death. So every single cell that we have in our bodies has a set of mechanisms that when they are induced, cause our cells to essentially go through the processes of self-killing. Mostly this involves perforating our membranes, allowing our mitochondria to start releasing a lot of factors that are going to activate a bunch of proteases, which cleave up all of our different proteins, and nucleases, which will eventually chop up our DNA. So it's a self-destruction mechanism. By inducing cells to undergo apoptosis versus something like necrosis or lysis, that's going to kill the cell in a contained way. So that's going to allow for all of those viruses inside of the cell to get killed along with it. The caspase cascade is that pathway of proteolytic activation. We activate many proteases that will go on and just essentially destroy the cell from the inside out. And of course, perforin is another protein that is going to compromise the integrity of the cell membrane, which causes it to bleb and really destroy its normal state. So this is a diagram just showing that. You can see our cytotoxic T cell here interacting with an infected cell. We have our MHC complex with the antigen presentation event occurring here. These cells, once they recognize the cell that they're compatible with, are going to start releasing factors onto that cell. In this case here, we see perforin. We can also have, again, that induction of apoptosis inside the cell to undergo that whole mechanism as well. But at the end of the day, the cell ends up dying as a result of this interaction. And once these T cells are activated, they can go around and detect many different cells that are showing this same signal, because any cells showing this signal is also infected and also needs to be destroyed. For our helper T cells, we have two major pathways that branch off of this. So these are cells, again, that are going to activate other cells to induce different mechanisms depending on what the context of the infection is, right? Again, that specialized immune response activation is what's happening here. We have the T helper 1 route and the T helper 2 route. The T helper 2 route is going to stimulate antibody production. The T helper 1 route is going to stimulate macrophages. So you can see here in this diagram, we have those dendritic cells we talked about last time that are at those common portals of entry, phagocytosing things, and then bringing them into our lymph nodes and other secondary lymphoid organs where our T cells are present. There's going to be that antigen presentation aspect that will occur. This helper T cell will detect this and then start to produce lots of different cytokines, again, cell communication proteins, that are going to either activate cytotoxic T cells or macrophages to go off and do that kind of work, or B cells, which is going to cause antibody secretion and other kinds of extracellular mechanism stuff. So if you want a way to remember the difference between helper T cells and cytotoxic T cells, it's pretty easy in their names. Helper T cells are going to just be helping the immune response to get started. They help other cells to become activated, whereas cytotoxic T cells specifically go around and kill infected cells. Cytotoxic, right? Cell killing. We'll jump over to the humoral response now, where we have lots of different things occurring for extracellular infections. We have antigens, which are foreign proteins or polysaccharides or other kinds of immunogenic factors that on their surfaces are going to have many epitopes. Epitopes are very small portions of an antigen that are recognized by an antibody. Remember that idea of the face, right? The face is a single antigen, but all of the parts of the face can be different epitopes that different antibodies can recognize. Antibodies are these Y-shaped protein molecules that are going to bind to one specific epitope. So those Y-shaped proteins have a very similar anatomy no matter what kind of antibody they are. They're all going to be Y-shaped. They all have 4 different proteins. So they have 2 light chains. These are these small blue proteins, and 2 heavy chains, which are these larger green ones, right? Heavier because they're bigger, lighter because they're smaller. And anyway, the light and the heavy chains make up 2 different binding sites that are identical. And the antigen binding site is going to bind onto that epitope that we talked about. So this diagram here shows this binding site blown up. So you can see this is part of the heavy chain. This is part of that light chain. And within, we have one small epitope. So let's say the eyes on the face or the nose on the face, whatever happens to be compatible with this particular antigen binding site that gets recognized by this antibody. If they're compatible, if they bind well, this antibody is going to stick to the surface of this antigen, and they act as an opsonin, which will trigger our immune cells to destroy whatever they're bound to. So every single antibody that exists is similarly structured, but is going to have a different antigen binding site. So imagine a bacterial cell with all of its different proteins embedded in its envelope, and all of those proteins having hundreds of different little sections on them, epitopes. We can have an antibody that will bind every single one of those epitopes and be able to attach to those bacteria and opsonize them. So this illustrates this very nicely. We have a bacterial cell here with many different, they call them here antigenic determinants. These can be epitopes, antigens, pretty much anything that's immunogenic. And they show them here as different shapes. You can see that different antibodies here have different affinities or compatibilities or specificities for these different antigenic determinants or epitopes. So you can have many different antibodies binding to the single surface of a bacterium based on what very small specific part of them that they might recognize. So you might be saying, well, how can we possibly anticipate every foreign protein? And how can our body respond to anything that might get into our system effectively? The way that this happens is very complicated, but every single naive B cell has a very different type of immune receptor on its surface. And those receptors can be swapped out, changed up, adapted at any time. And there's always new B cells that are getting produced, right? So we have this seemingly endless number of possibilities of receptors on these cells that can detect virtually anything just by swapping out even a single amino acid in a codon or changing up a large portion of a gene through cut and pasting mechanisms that are embedded in the genes that code for these proteins. So there's a ton of versatility that exists there. And so as these cells are in constant circulation in our bodies, eventually when they do run into whatever they're compatible for, then they get activated. Following activation, they're going to be able to do their effector function, but they also proliferate. So you might have 1,000 B cells that are naive, that are in circulation, and you might have maybe 5 of those that are compatible with the bacterium that you're infected with. And so when they bind that, they're going to start to become activated, and they're going to double, triple, quadruple, keep dividing and dividing to make this massive army of a very specific subset of cells that can match that pathogen and fight it effectively. So this is shown well here, where we've got our repertoire of B cells that are just in circulation, and the antigen that exists for whatever pathogen is infecting us at that time is only recognized by C. So this population is going to be formed through that division of these cells. So we're going to get a large number of effector cells that are going to get produced as a result of that initial antigen recognition and activation. Another population of cells that's going to form are our memory cells. Remember, we've been talking about immune memory. So not only are cells going out to fight, but we have another set of cells that just persist in our body after this initial infection is cleared, and they're looking for this infection again. So if it happens to come back, comes around the back door next time maybe, instead of knocking on the front door, or tries to come in through the window, these cells are ready for it. So following their activation, effector B cells are called plasma B cells. They just change names. So naive B cells have never seen antigen. Once they see it and become activated, they become plasma B cells. And then, of course, we have our memory cells that don't fight, but just kind of hang out, and our cells that are kind of mature B cells that have all of the refinements done to their proteins that make them the biggest and the best effector cells that can come back and fight that infection again if it shows up again later. So here's kind of this whole humoral immune system process from start to finish. We have our bacterium here that is getting engulfed by a cell, an antigen presenting cell. It's going to show that to helper T cells. Helper T cells, through antigen presentation, recognize that there's an infection present. They start to become activated, produce signals that can then activate B cells who recognize this same microbe. And then after that, those B cells are going to differentiate, proliferate, divide. They're going to turn into plasma cells that are active antibody secreting cells that are our effector cells of the immune system. And then we've got also these memory B cells that are going to persist in circulation. One quick side note here, B cells, because of the vast amount of proteins that they produce, every single thing that they are making and secreting is protein in nature, they have a huge endoplasmic reticulum. If you've forgotten, the ER is the location in the cell that is responsible for making protein. So when you look at the ER, it pretty much takes up the whole cell because they don't really have another function or purpose other than producing antibodies in an immune response. So that's pretty neat that you can see that with electron microscopy. So sometimes I like to give you several diagrams of these same kinds of complex mechanisms just to help or give you different ways to look at the same picture. But this essentially shows what we've just discussed. Engulfment of a microbe by an antigen presenting cell, presentation to helper T cells with that MHC complex. We have activation of the T cell, which then can in turn activate B cells, which can become plasma cells, which can then start to produce and secrete antibodies. What's neat about B cells is that they don't need this piddly antigen presentation step in order to detect antigen. They can sense those whole organisms that are floating on by, that are all huddled in the corner plotting their next big event in our bodies. And that makes them a little bit more versatile than our T cells that just kind of sit and wait to have things shown to them. The effects of B cells, because of how they work, and by the fact that they secrete things into our bloodstream, really makes them highly effective body-wide. So if we only have a couple of B cells that are proliferating into massive numbers, And then those antibody producing cells are making tons of antibody and throwing it into the blood. No matter where that infection is, even if it's spreading around, those antibodies will find and reach them. To contrast that, our T cells only work in very close proximity with the infectious agent, and only when it's been degraded and shown to them. So, two very different mechanisms that achieve a similar thing. So when we think of antibodies and their effects, there are many different ones. We can have neutralization, where antibodies will simply bind to surface receptors and cause them to become ineffective. If we have something that is blocking or impacting the ability of things to bind to a pathogen surface, it's going to really decrease the effectiveness of those surface binding proteins. We can also have agglutination or precipitation, where many antibodies will bind many different bacteria or their toxins and cause them to clump together. And then those large complexes that are formed can get targeted and cleared by our phagocytic immune cells. Opsonization is another one we've talked about a few times already, but again, to target and signal to our immune system through binding that something is foreign and needs to be degraded. And antibodies will also activate the complement system, which we talked about last time as part of the innate immune response. And this is a great summary diagram just to run through all of the things that we just mentioned on that previous slide. Immunological memory, once again, follows that primary exposure of the immune system to an antigen. So after we have had the opportunity to determine the type of infection, which part of the immune system needs to be activated, refining the proteins, receptors, antibodies, etc., to make them the most effective that they can possibly be to fight that pathogen, then whenever we have a secondary exposure, the clearing of those infections will be more rapid and on a much larger scale. And that is that adaptive nature of the adaptive immune response. We learn every single time we get an infection, and we retain that knowledge just in case those come back around again. Things like antibody titers are a great way to determine whether or not you've had an infection with something previously. This is the amount of antibody against a certain antigen that's present in your blood. And if you do have antibody in your blood, that would indicate that you've had an infection with a pathogen that antibody is specific for. It's a direct measure of how intense of an infection that you've had based on how many B cells have been activated and how much antibody your body has made against an infectious agent. This is easily quantifiable with tests. So if there's ever a time when you need to make sure that you haven't had a certain kind of disease, like tuberculosis, for example, they can do an antibody test of some kind. So looking at this diagram here, you can see antibody titer. So the amount of immunity, if you want to put it that way, is on our y-axis. And then our time in days here is on the x-axis. So let's say we have an initial exposure to a pathogen. We're going to go through those motions of producing that immune response. We're going to have an increase of antibody production here. You'll see that as that infection is cleared over time, these amounts are going to come back down. But if we get a secondary infection, we're going to have this huge spike, right? Because we have those memory cells that are in place, and they can come around and produce antibody in much higher amounts. You might be wondering here what IgG and IgM are. It's not so important to our purposes here, but IgM is more or less this pentameric, giant, clunky antibody type that's only really used the first time that your body gets an infection. It's used mostly inside of the bloodstream. And in a secondary infection, you'll see it's not really activated much at all because it's not a very effective antibody. IgG is another type that's much smaller. It can actually pass outside of the blood vessels into different interstitial tissues. There's a whole bunch of different antibody types. It's not so important, but this is just showing again that our secondary immune responses tend to be bigger, more effective. So this is a nice complex diagram showing something very similar where we've got naive B cells that are first exposed to antigen. You can see that they're going to start to produce different kinds of antibodies over time. And then after a while, we get that memory cell population that gets produced. These are going to have better, more refined antibody types to go and fight the type of infection, whether it's going to be in the blood, whether it's going to have to cross the placental barrier, whether it's going to be at the epithelial layer. There's all different types of antibodies or derivatives of them that act at different parts of the body. There's 5 of them. We won't talk about those in too much detail. But what we're seeing here, after that secondary infection, that memory population is going to be producing antibodies in larger numbers, more specific types of antibody that's more effective, and that's going to be something that helps us to fight those infections better. And just for a little aside here, when we get a vaccine, our vaccines cause this response, right? That's our primary exposure to an infection. But this is a killed infection. Probably we have vaccines that are attenuated versions. We have these mRNA vaccines that will produce certain epitopes of a virus or other types of organisms for our body to identify and mount an immune response against. So our B cells in this case still detect the parts of the vaccine as if they were a living organism, right? They're still foreign parts of something that could be foreign and infectious that we haven't seen yet. And then we mount our immune response to it. This is so that if we ever get the infection, for real, that we have this immune memory already in place. So if we were to get, for example, a vaccine against COVID, we're forming our primary immune response against those spike proteins or whatever the target happens to be. And then over time, if we get infected with COVID down the line, we've already got an immune response ready. So we can fight that infection better. This will limit the number of people who end up with severe versions of the disease. For all of the reasons that we talked about, when we have people that are not necessarily having that infection present in them for as long, so the virus doesn't have as much time to cause damage, to cause long-term effects, because it's getting cleared faster than it would if we had never had that vaccination. If the first time that we see COVID is this, our body is going to be a lot less effective at clearing that infection because it has all of that learning to do. So getting any vaccination sets us up for success when we actually run into the real thing. We'll switch gears now. We'll talk about antibody-dependent cell-mediated cytotoxicity. And this is a situation that occurs when pathogens are too large to be phagocytosed. We kind of talked about that a little bit last time, where we have large worms or other multicellular parasites that cannot simply be engulfed. And we talked about how cells will start to secrete many degradative factors out towards those organisms to break them down and destroy them. Antibodies are actually a large part of this process because when they get produced and secreted, they'll bind to these organisms and help to draw chemotactically a lot of our destructive immune cells to that area to start firing. So antibodies are in many ways that initial signal to our immune system that something is wrong and work needs to be done to get rid of something. You can see that here where we've got a nice large parasite that has all of its epitopes on its surface, and our B cells have produced antibodies that have stuck to that surface to signal to our immune system that that worm needs to be destroyed. So you'll see here that that recruitment effort has gone over well, and we've got lots of cells. Eosinophils are a type of cells that we talked about last time. They show up with their granules and start to secrete them. Our macrophages come along and start pulling these things apart piece by piece. And over time, we end up with those pathogens being destroyed. You can see over here in some electron microscopy, a very large parasite that is just swarmed with eosinophils as they're breaking it down to destroy it. So basically here, this is a nice summary showing the differences between the humoral or antibody-mediated response and our cell-mediated response and all of the different steps that take place. We won't spend too much time here, but it's just, again, to kind of wrap up this little side topic quite nicely. We'll talk now just briefly about vaccines, which are again an example of artificial immunity. And vaccines can be made from all different types of things. They can be inactivated toxins, killed microorganisms in whole form. They can be living but attenuated microorganisms, meaning that they're still alive, but they can't replicate or induce that toxicity that they normally could if they were present in their normal state. Or they can be microbial parts of microorganisms like capsules, or in the case of these mRNA vaccines, where our body is producing different types of factors that would normally be present on the surface of a pathogenic organism instead. Even in some cases, the DNA of microorganisms is possible to go into a vaccine as well. So these are some great examples of vaccines that are used in the United States to prevent bacterial diseases in humans. Again, not a table that you need to memorize or really spend too much time on, but it's just showing you the different types of things that are going into these vaccines in terms of what portions of microorganisms or their toxins that get added to these in order to activate our immune response against them. We have another table here just showing some viral diseases. And again, we won't spend too much time here, but just to show you what's actually going into these, when they are administered, and how often they should be administered. We've already talked quite a bit about antibodies in research, and now that we have a better understanding of the immune system, maybe some of that information will make a bit more sense But in the research lab, when we raise antibodies, we can raise them against any kind of protein and detect that in a way that is helpful to us for diagnostics or even for developing therapies for people. This again shows us how to make an antibody where we can inject any kind of antigen with all of its epitopes, and then we can raise those B cells that are going to produce those antibodies en masse. We can fuse them with other cells to create a hybrid cell that will be more long-lived than B cells on their own. Primary cells, again, only have a certain number of divisions that they can undergo before they die. By fusing them with a cell that is more long-lived, like a cancer cell, which does not have a lot of cell division regulatory mechanisms in place, we can elongate the lifespan of that cell by a large measure. And then what we can do after that is collect all of these cells that produce these antibodies, grow them up in culture, and at the end of the day, get ourselves a nice antibody concoction that we can then use in the lab against whatever this initial target was that was injected in the first place. Blood typing is another really excellent way that we've used antibodies that we're probably a little bit more familiar with, because if you recall, each one of our red blood cells can have different antigens expressed on the surface of those. So when we want to determine the blood type, whenever we see antibodies binding antigen on the surface of blood cells, they'll clump together, and then we can determine their blood type. But we won't spend too much time on this because we've talked about this a lot in other courses already. Enzyme-linked immunosorbent assays we've already talked about before as well. But again, in the lab, when we want to study a target protein, whether or not it's present, how much amounts it's present in a certain cell in given situations, we can obviously tailor antibodies that are attached to some kind of indicator molecule that can be light or color or whatever, and that will allow us to detect the presence or the amount of something that is in a sample. A couple of other little topics to touch upon. Immune deficiencies or immunodeficiencies can occur in individuals from when they're born, or that state can get induced later in life as well. Babies are born without broad immune systems, and they obtain antibodies and develop immunity in utero and through breastfeeding. And obviously, as babies start to interact with their environment, they come into contact with different microorganisms throughout their young lives. They start to develop their own immunity over time. There's also the idea of immunosenescence. So after you hit the age of 50, typically your immune system starts to become less active. You become less effective against combating infections, and this is in part due to your actual thymus slowly turning into fat. So your thymus is not as robust in the later years in life as it is when you're younger. But apart from this, individuals can be born without portions of their immune system that make them predisposed to infections or not being able to fight them off as effectively. Autoimmune diseases also exist, where all of these mechanisms that we've talked about today that are meant to target and destroy foreign pathogens actually accidentally turn on your own tissues and target and destroy those instead. So sometimes your system will act out of turn if you have a certain kind of disease state or some kind of issue, and that will induce your immune system to react against your own tissue. Auto is usually self, and an autoimmune response is an immune response against your own tissues. There are treatments for this, usually though they're generalized immunosuppressants that just kind of dampen down the entire immune response. You can never really turn the entire immune system off. But by suppressing the immune system, it does make you less effective at fighting normal infections, but it also decreases the effects that your immune system is having on your own tissues. Hypersensitivity disorders are broadly classified as different kinds of allergies, and again, this is a misplaced immune response against stuff that isn't actually a threat to you. Don't forget your immune response is in place to protect you. But sometimes, if you sniff a flower and your body decides that that flower is immunogenic, you can just be allergic to every flower that you ever smell after that. And a lot of hypersensitivity disorders that can arise from misplaced immune response activation are things like asthma, anaphylaxis, hay fever, food allergies, and any other number of things that come to mind when you think of allergic reactions. There are mechanisms to help to combat allergies, and immunotherapy is one of them. Immunotherapy is actually an option for many hypersensitivity disorders where you get a regular injection with something that you consider to be antigenic to develop tolerance against it. Sometimes this works really well for people. Sometimes it does not. So it's really just a gamble of trying out something new to see if it will be effective for you and if you can eventually stop avoiding things that cause you to have an allergic reaction. So you receive injections over the course of several years usually, and at the end of that, you may or may not be immune to whatever used to give you allergies. This image here is showing a hypersensitivity test where they have the scratch test on the skin. So they'll have a number of different things that they test you for, and they'll kind of just scratch the surface of your skin and add a concentrated amount of an antigen, so maybe common allergens. So they might test you for cats, dogs, grasses, birch. These are all the things I'm allergic to that are coming to mind. And if you do have an allergy to them, you're going to have that hypersensitivity reaction wherever that was applied to your skin. So if you see this red bump that forms, that means that you're allergic to whatever they've applied to you. And then they can make you a long grocery list of all of the things that you need to avoid in your life. Or potentially make you a concoction of allergens to try to develop some tolerance to some of these things. There's also the idea of organ and tissue transplants, which is a big topic in immunology. And you can have transfers of organs or tissues between two members of the same species. That's called an allograft. You can have transfer of skin or tissue from one part or another from the same person as well, which is called an autograft. And when we talk about rejection, where you get some kind of transplant, but your body rejects it, that's usually due to these immune mechanisms that we talked about today. When you are giving some kind of transplant to another person, you have to make sure that there is compatibility between those people. So when they talk about finding a match from a database, that match has to be molecularly compatible. And there are many different checkboxes that you can go through to compare two people's proteins and other factors to see if they would be compatible with one another. The worst thing that you would want to go through is giving somebody a kidney, and then that kidney gets rejected by their body and destroyed because their immune system thinks that it's foreign. And just to touch on this briefly too, cancer is something that's not easily combated by the immune system because the disease arises from host cells. That's not to say that our body doesn't realize that cancer is there and does not try to fight it. It's just difficult initially to detect. Cancer cells can look mostly like us, but might have maybe too many receptors on their cell surface, or might grow a little bit too fast. So while our bodies can kind of sense that there's something the matter, sometimes it's difficult to actually mount a response and kill cancer cells before they become established. But we have many other mechanisms in the body to eliminate cancer cells. The immune system does play a part in it, but again, because they are our cells that have become dysregulated in a way, our immune system is not as effective at combating them as it would be something completely non-self or foreign. And if you found any of these topics interesting, of course, as I mentioned last time, I do teach a third-year course in immunology, and we will go into a lot of these couple of topics that we just kind of breezed over in great depth in that course if this is something that you want to explore further at some point. But as always, if you do have any questions, you know where to reach me at laura.r@nipissingU.ca. Thank you so much for listening, and I'll catch you all soon. Bye for now.